This video is about foundational concept number 1, Biology 1A, proteins and amino acid structure and function. This is the first video about protein structure. So we're talking about the primary structure of proteins, secondary, tertiary, and quaternary structure of proteins, along with some important amino acids. The primary structure of protein is present in every protein, and it is just the string of amino acids that are linked together with peptide bonds. It's read from the N terminus to the C terminus. The secondary structure of proteins includes the backbone interactions such as hydrogen bonding between the hydrogen of the nitrogen and of one amino acid and the carbonyl oxygen's lone pair on another amino acid. The two most common secondary structures are alpha helixes and beta pleated sheets. The alpha helix is quite stable because there are many hydrogen bonds formed in this arrangement. The R groups do not contribute to the hydrogen bonding that forms this alpha helix. And transmembrane proteins have alpha helices that span the membrane for the transportation of ions into the protein. Beta sheets are also formed through hydrogen bond interactions and are aligned in rows. There are two types known as parallel and antiparallel sheets. Parallel sheets have adjacent polypeptide chains lying in the same direction, nitrogen to carbon, and antiparallel sheets have adjacent polypeptides lying in the opposite direction. So one is nitrogen to carbon and the other is carbon to nitrogen. The tertiary structure of proteins is essentially the 3D structure of proteins and is created via electrostatic side interactions of the R groups. So the positive charged side chains, such as histidine, lysine, and arginine, interact with negative side chains, so aspartate and glutamate. Polar side chains are attracted to other polar amino acids, and nonpolar side chains are attracted to other nonpolar amino acids. So we talked about before, cysteine creates covalent disulfide bonds between two different cysteines, which join together to form a bridge connecting parts of the protein. So the cysteine dimer is called cysteine without an E, and it functions to stabilize the protein's final conformation. So a protein that has a lot of cysteine in it is generally more stable than a protein without a lot of cysteine in it. Proline has a unique cyclic side chain which reconnects back to the amino acid backbone. This has two consequences, giving the protein conformational rigidity and making the protein in this area unable to hydrogen bond because there's a lack of available hydrogen when proline is part of a polypeptide. So proline destabilizes the secondary structures, creating kinks, which are useful to accommodate turns and for protein folding. Glycine is another important amino acid in tertiary protein structure. Its side chain is a single hydrogen atom, so this makes it the least sterically hindered amino acid. Accordingly, it can easily rotate and move, and it is often found in protein areas which need flexibility and rotation. The last type of interactions that occur with the tertiary protein structure are hydrophobic interactions. So hydrophobic bonding brings together hydrophobic parts of the polypeptide by pulling them into the protein's interior to protect them from an aqueous environment, which just means an environment that has water. So this occurs between the hydrophobic amino acids. So just as a quick reminder, the hydrophobic amino acids include valine, leucine, isoleucine, proline, phenylalanine, methionine, and tryptophan, along with glycine and alanine to a smaller extent. This removal of the nonpolar groups from water is known as the hydrophobic effect and is the primary force that stabilizes tertiary structures. So we're at the last structure of proteins, which is known as the quaternary structure. It's important to note that not all proteins have this structure. It occurs when interactions between two different protein subunits, for example, two separate polypeptides, which can be either backbone or side chain interactions. So for an example, covalent disulfide bonds formed between two cysteine residues contribute to a quaternary structure. So proteins with a quaternary structure can be known as dimers when there are two polypeptides linked together, trimers when there are three subunits linked together, tetramers when there are four subunits linked together, pentamers for five, etc. Hemoglobin is a popular tetramer protein that is going to be tested on the MCAT, which we'll be talking about in later videos. The quaternary structure is formed after the protein has been translated from mRNA. So some post-translational modifications are part of the quaternary protein structure. So they include phosphorylation at certain amino acid residues, such as serine, threonine, tyrosine, aspartate, and glutamate. Disulfide bond formation between cysteine residues, creating cysteine without an E. Glycosylation, which is the addition of a short carbohydrate polymer to the protein. And lastly, ubiquitination, which is when ubiquitin, a protein, attaches to another protein to mark it for degradation by our bodies.